Hello, my name is Gabriel Dunn. Today's date is July 22nd, 2018. In this week 3 video lab for microbiology, I will be discussing and demonstrating different staining techniques. The first exercise in our lab this week, it was a direct stain of the cheek cells. So we did that in a different lab exercise without staining it, and it was very difficult to see the cells on the slide because of that. So this time we're going to go ahead and do some staining. We're going to do a direct stain with the crystal violet, as well as the negative stain and the differential stain, also known as the gram stain. So here I've already taken a swab, sterile swab, and I swabbed the inside of my mouth and my cheek with this, and I placed it on this slide here. And the slide was then heat fixed with this candle here. The manual said do it about 5 times, not to overdo it and kill or destroy your samples, and then to let it sit for approximately 10 minutes. So I've done that. Now I'm going to go ahead and apply the stain here. So I'm going to put the camera down, do that. Add a drop or two. To that. Gonna let it sit there on the slide specimen for approximately 60 seconds. After which I will wash the dye off with distilled water. So the crystal violet is a basic dye, and it works based on the electrochemistry of the cell wall of the cells that we're looking at. It's slightly negative, and so the crystal violet has to be opposite charge because it's going to directly stain the cells. So it has to be an opposite charge. That's the way it is able to dye the cell. The negative stain is a little different. It's going to repel the charge of the cell membrane, and so it's not going to be able to stain directly. It's going to stain around the cell, so it's going to kind of make an outline of it, but not stain it directly. Okay, so this is now ready to be washed out with water. So we're gonna take that and just wash that. Okay, no more purple dye. It's clear water. And I'm just going to pat this dry with a paper towel. I'll just put it there. See, don't wipe it because you'll wipe off the smear and that'll be no good, so just gonna pat it dry. And then here's my direct stain. And we can look at that under the microscope, take our pictures and make our observations. Also, I'm going to do the negative stain Congo red with the same sample, with another swab. This time it's going to stain the background, so that's going to be interesting to see. I'm just going to put one drop here, then I'm going to put my sample, then I'm going to take another slide and just smear it down at a 30-degree angle all the way down the slide, and then let that dry and take a look at that. And then also I have the 3 other slides we're gonna make here using the gram staining techniques, using the broth of these 3 microbes that we had cultured in week 1 and week 2. And gram staining, we're going to use 4 steps to do that, and then observe the results on the microscope. Alright, that's going to go ahead and do it for my video. Thank you so much for watching.